Hello, everyone, and welcome to today's DDN webinar. My name is Yuning. I'm the Assistant Science Editor for DDN, and I'll be moderating our discussion. We have an exciting webinar planned for you today. Our speaker, Arutha Kulasinghe, will discuss how his team uses spatial transcriptomics to uncover the mechanisms of SARS-CoV-2-induced effects on multiple organs and tissues. After the talk, Dr. Kulasinghe will participate in a live Q&A session. If you have any questions or comments during the talk, you can simply submit them to the Q&A portal to the right of your screen. We'll try to get to as many of these as possible. I'd like to take this opportunity to thank our webinar sponsors, Sinobiological, Canopy Biosciences, 10x Genomics, and Fortis Life Sciences. Our sponsor has provided us with some helpful handouts related to today's webinar. You can find and access these in our handout section, located on the right side of your screen, where you can also find your certificate of attendance for participating in today's live event. And with that, let me introduce our speaker, Arutha Kulasinghe. Arutha Kulasinghe is a Peter Doherty National Health and Medical Research Council Research Fellow and leads the Clinical Omics Lab at the University of Queensland. Kulasinghe has pioneered spatial transcriptomics using digital spatial profiling approaches in the Asia-Pacific region, contributing to world-first studies for lung cancer, head and neck cancer, and COVID-19. He aims to understand the underlying pathobiology of these diseases using an integrative multi-omics approach. Let's just take a minute to make sure your slides are working smoothly. Dr. Kulasinghe, please take it away.
Thanks, Yuning, for the invitation. Can you hear me okay?
Yes.
Yeah, perfect. Okay. Thank you for the invitation, and then lovely to be on this webinar. So today I'll talk to you about developing a spatial organ atlas for COVID-19. A research lead at the Fraser Institute at the University of Queensland in sunny Brisbane, and we've acknowledged a number of funding agencies that support this work. My talk today will triage from what we're doing in the cancer space and how we've applied that to looking at the impact of COVID-19 on multiple organs. It was a serendipitous we had developed the workflow, and then so the right tissue types were available at the time we could really apply that. So hopefully we'll get some insights into that today. So we're based at the Fraser Institute at the Translational Research Institute in Brisbane. This is a 1,000-person building that's located on the campus of a hospital. And so we do a lot of translational and clinical studies at this location. So today I'll be talking to you about spatial transcriptomics and how we've leveraged that to really understand the intricate biology that's present in cells and tissues and organs and so on. And so when we look back, sort of bulk genomics, this fruit salad analogy is probably overused at this stage, but it sort of gives you an averaged expression count for, all your transcripts that you've looked at. I don't really have an appreciation for lowly expressed or highly expressed. And it sort of averages out your signal. In 2011, with the advent of single cell sequencing, we could sort of, order these cells. We knew how much of an individual cell type there were, but we didn't really understand where these were. We could project this onto a t-SNE and look at, identifying cell populations and so on, but we didn't really appreciate how these exist in organs and in tissues and understand the complexity of this. And so what's happened over the last 4 to 5 years is this advent of spatial transcriptomics has really enabled us to see the cells in the context in which they're found in the body and in the organ and in the tissues. So we don't need to necessarily dissociate these cells and sort them. We can look at them as they are in the tissue. And that's powerful because we can start to understand the cellular content, the cellular localization, the certain neighborhoods that are found in the tissue, and so on. So my lab does a lot of, almost exclusively spatial sort of profiling, biology, transcriptomics, proteomics, and we're layering this omics stack, but very much focused on spatial. And so spatial biology is giving us new insights into disease biology. We've written a couple of pieces for Gen-Eng Biotech, how spatials are reorientating the biopharma development, how it's redefining the multi-omics approach. And we've had a couple of papers, which I'll talk to you about a bit later, on the impact of understanding viral localization in the lungs and in the heart, and the associated sort of profiles we were able to look at in these studies. I'll initially focus on the cancer work just to give you context into the approach, and then we'll go on to the COVID-19 studies. So in the immunotherapy field, we know that there are predictive biomarkers, currently limited tumor mutation burden, which is sort of a bulk expression analysis of the tissue, or PD-L1 expression. And so the challenge is these are bulk measurements of the tumor microenvironment, and you're not really able to understand the intricacies of the tumor microenvironment. And so what my lab does is try to understand how these cells communicate with each other, how they communicate with their immune cell subtypes and neighbors in the tumor microenvironment. And we try to profile these cells almost cell by cell in the whole tissue. A typical slide will contain 2, 3, 4 million cells. What we try to do is annotate these and then look at the associations with clinical endpoints. And so why spatial is important, if we look at these two tissues on the bottom left, if we were to mash them up and look at a bulk expression score, this is called the TIS score in this instance, tumor infiltrating score, they would have very similar TIS scores. But the tumor on the right is highly infiltrated compared to the one on the left. You can see that you've just got more pink cells within the tumor mass in the green. And so getting that visual imaging data and understanding where that is in space and in the tissue is important to, predicting response to therapies in this instance. And so a bulk measurement doesn't give you that. There is a caveat though, spatial does take a village. So we use a number of approaches. Initially, our lab was doing unbiased whole transcriptome type approaches using a number of methodologies listed on the left here. And then we typically go and validate a lot of these findings using spatial proteomics. And I've named a couple of technologies we use here. And so how this has evolved over the last couple of years is there's been this shift as the plex has increased on the protein end. We are starting to do more discovery work from spatial proteomics, and that's really powerful because that lends itself into sort of the pathology space, digital pathology space, and it's just a more robust assay for the clinic. And so how this comes together is this really challenging piece. We call it sort of the spatial informatics or computational biology, where it's bioinformatics, biostatistics, imaging, companies, spatial informatics teams that we work with AI companies as well to really understand the complexities of these data sets. You're often working with a single experiment could be 10 terabyte file size. So these aren't sort of, run-of-the-mill assays. So getting, having bioinformaticians, data scientists, maths, single molecule fish, ecology experts on these projects is critical. So this is how we've developed that sort of expertise within the team. And so I'll talk about some of the work in cancer, but we'll triage to COVID-19, but it shows you how we can use this approach to understand predictive biomarkers for response to therapy and so on. So this is a tissue microarray where we had a single biopsy, core biopsy per patient. 45 patients were treated with immunotherapy, 45 were treated with standard of care, platinum therapy and lung cancer, and we had a single biopsy per patient sample. And so we used a gating strategy of masking on cytokeratin for the tumor, and we didn't want to be selective and go and look at CD45 or a sort of a immune cell component. We gated on non-cytokeratin to effectively get the tumor microenvironment. And so for each patient sample, you've got representation of tumor and what the microenvironment or stroma signal is. And then we could look at how these parameters measured at RNA using an 1800-plex assay and then a protein 60-plex assay associated with response and resistance to therapy. And some of the IO cohort details are on the right there. And so this is now looking at the enrichment of the proteins and the genes, the mRNA transcripts between the responders in the left and the non-responders on the right. Those mRNA targets and protein markers that are significantly differentially expressed between response and non-response are in the blue. And where it got interesting for us is we can take these and look at them both in the tumor content and the stromal content. We could see that in the stroma, SPP1, osteopontin, and IL-2 were highly enriched for in the responder group. And when we look at the contribution of IL-2 in a multivariate analysis, IL-2 almost by itself predicted the patient cohort that would benefit from immunotherapy. This is IL-2 specifically in the stromal component. And so you can see, using an unbiased lens, how you can leverage spatial to potentially identify new targets that associate with response, and then potentially go validate these findings in independent cohorts. And so we took that same tissue approach and plexed it, that same tissue microarray, and we plexed it at 6 markers, looking at PD1, PDL1, FOXP3, CD8, 60, CD68, and cytokeratin. And we could look at the composition of each of the cores based on these markers. This is with single cell readout for about 7 different proteins. And we can look at the co-expression of markers as you moved into the tumor and as you moved into the microenvironment. And so when we've annotated it using this 6 or 7 plex assay, we can then identify the cellular neighborhoods that are present within each tissue microarray core. So a cellular neighborhood is saying that it's using a k-nearest neighbor analysis. It's saying that my tumor cell might be next to a CD68 cell, might be next to a FOXP3 expressing cell, and so on. We identified each individual neighborhood within each individual patient sample or core, and then we extrapolated it to the 90 patient cohort. And we could ask the question, what was the enrichment of the neighborhoods between the patients that responded to therapy versus those that didn't respond to therapy? And so we could see this CD68, PD1, FOXP3 cell interaction that was highly enriched for in the non-response group. Obviously, at 6-plex, we are binning a lot of cells as other, and so your cellular neighborhood gets a lot better as your plex increases. And so we followed this up with a 36-plex assay to resolve this down a bit better. This works in prep. Should be submitted quite soon. And so we could, based on 35 markers, we could characterize multiple additional cell types within the tumor microenvironment of this tissue microarray, where each column here represents an individual core, and this is just breaking it down between patients that responded to therapy versus those that didn't. And we could see this association of the presence of an increased presence of Tregs, specifically in the non-response group. And so what we see here is we've taken the multiplex IF data on the top, and then we've identified the cellular neighborhoods within those cores once again at the bottom. And then we could ask the question, what is the neighborhood enrichment between the response and the non-response group? And you can see these, on the volcano plot on the bottom right. You can see those in the red are the significantly enriched neighborhoods between the patients that responded versus those that didn't respond to therapy. And so then we can take this graph neural network approach and then look at the nearest neighbor in this type of analysis. And we could see that the Tregs and monocytes, or the HLA-DR tumor cells and macrophages, or the Tregs and CD8 cell proximity, when you had those cell types in proximity, they were associated with response or resistance to therapy. And we are trying to get a bit more granular on this and sort of develop spatial scores as a function of distance to or from these cells. And this is linking to a lot of the work that was done by the Nolan lab. But we're using it as a workflow to really demonstrate sort of the proof of principle in our study. And so we've taken these tissues and we've multiplexed them, whole tissues. This is about 3 million cells at 57 colors with single cell readout. And so you can get an appreciation for, tissue lymphoid structures, lymphoid aggregates, and so on in the CD20 channel here in lung cancer. But what we saw in the study was that there was a high, and this is in a cohort of patients that had that were given immunotherapy, and we had their clinical endpoints and response to therapy data, and we could see that there was this increased expression for metabolic markers, specifically in the tumor compartments of those patients that did really poorly. So what you're seeing here is increased presence of glucose uptake, specifically in the tumor regions of the patients that did really poorly. And what we're trying to really drill down on in the lab at the moment is, can we develop a minimal signature predictive of resistance based on a metabolic score? And so in patients that do really well, we see a flattened sort of metabolic activity. And so we're digging into this literature a lot more. And so we can develop sort of cellular neighborhood maps of what, a pathological complete response looks like versus a stable disease and so on. And so we'll go on to this really exciting case. It's an index sort of study that we've run, really showcasing the power of spatial. And this is over 103 antibodies stained on a single tissue section in head and neck cancer, HPV positive disease. This patient died after 14 months. We had the pre-IO specimen. And so we knew that this patient would inherently have resistance in within their tissue. And so we went in trying to find out whether we could identify it. So this is an H&E section, and we can look at the cellular architecture directly on the tissue using these spatial approaches. And so where it got interesting for us is we can do the multiplex IF imaging, but then based on the co-expression of over 100 markers, we can now cell annotate them into this sort of atlas. And what you see in the yellow and red in the middle is normal tonsil, but what you see at the top right is a tertiary lymphoid structure specifically infiltrating that bit of the tumor at the top right. These TLSs are absent in the top of the tumor and the bottom. And so we can break this tumor down into subtumors, and we can see that these sort of tertiary lymphoid structures exist in tumor region 4 almost exclusively. And so when we know that these tertiary lymphoid structures sustain an immune responsive microenvironment, there's a lot of literature showing this. And so we can, this is what we see in this patient specifically in that case. And when we look at the localization of immune cell subtypes, we can see that your M1 macrophages are localizing to this bit in the yellow, the area where you have your TLSs. The M2 macrophages are in the green, blue, and purple region. So the M2s generally are immune suppressive. And so on 2 dimensions, you can start to appreciate this partial resistance to IO therapy. One being the presence of TLSs specifically in one area of the tumor, but absent in the other areas, but also this M1, M2 macrophage localization. And so we looked at sort of broad markers that pathologists look at. CD68 in the green, it's highly infiltrated. You might say there's more at the top than the bottom. CD8, again, highly infiltrated, probably more at the top than the bottom. But what was really powerful to see was when we looked at the metabolic data on this tissue. And so what we could see was glucose 6-phosphate dehydrogenase was enriched for in tumor region 3. That's the area that lacks TLSs. That's the area that has the M2 macrophage phenotype. It's a highly proliferative and hypoxic region. So that's your resistance to therapy. That's the area that will likely lead to the sort of resistant phenotype in this patient. Whereas we saw BAX and a few pro-apoptotic markers enriched for in the green in tumor regions 1, 2, and 4. So all of tumor region 2, 1, and 4, where you have your TLSs and one macrophage phenotype is all dying. It's responsive to therapy, whereas the tumor region 3 is what's likely to be resistant. And so we developed a spatial metabolic map of this tissue, and we could see the pentose phosphate pathway was up in tumor region 3, bordered by fatty acid metabolism and DNA damage. And we're digging into this a lot deeper because we think that might be the sort of invading edge of the tumor. And so tumor regions 2, 1, and 4 are high for all your sort of pro-apoptotic pathways. So now that's sort of a really nice segue into the COVID-19 work. This was from the Hopkins data base. And, this was taken in November last year. It's just, it's affected us daily. We know that there've been, almost 7 million deaths because of this virus. So we were trying to dig into really understanding, using a lens of spatial, could we understand viral localization in the lungs and the potential impact on multiple other organs. And so this paper came out in, I think it was April 2020, and it was a bulk gene expression study that essentially had this Venn diagram in it. And it showed that, COVID versus flu, the 2009 pandemic, there were a number of differentially expressed genes, but it didn't really give you an appreciation of where the signal was coming from. And it sort of piqued our interest into this. And so we published a number of studies, which I'll go into now, looking at the impact of COVID-19 on the lungs, the heart, the placenta, and we've got ongoing studies looking at the brains of COVID-19 patients taken at rapid autopsy. And so this was sort of the study design. We had tissue samples collected from Curitiba in Brazil. At the time, these were all unvaccinated patients really early on in the pandemic. We were looking at lungs, hearts, brains, and placentas. And we had the 2009 H1N1 tissues as a comparator and normal control non-viral tissues as well. These were prepared into tissue microarrays. So an example is on the right there, 10 patient samples, core biopsies of the lungs, H1N1. And we try to match these as much as possible for age, gender, and so on, and normal as well. And so we had these pathology annotated by local pathologists at the time in Australia, sort of mid-2020. We didn't really have a caseload, and so none of the pathologists had seen COVID-19 lungs at the time, but they were in touch with the literature. So there were a number of really key collaborators that came on the project. We stained the tissues with Mason's trichome for fibrosis, IHC for the spike, and RNA-scope for the spike as well. And TMPRSS2 and ACE2. And at the time, we ran the NanoString Geomics DSP COVID-19 Immune Atlas panel, which was built off their Cancer Atlas panel. But since then, we've also run the whole transcriptome over these samples. We took the tissue-based findings, and then in a pre-Omicron study, we looked at those genes in the blood and nasal swabs in about 1,000 patients in a cross-sectional multi-cohort study, again, pre-Omicron, where it wasn't rampant across the world. And so we could localize the virus in the tissues using RNA-scope. This is a depiction of it on the bottom right. The areas in red are tagging where the viral infections are in the lungs. And so we could identify regions within the tissue that we wanted to profile based on that. So under the guidance of a pathologist, we could identify the hyaline membranes, the type 2 pneumocytes, the bronchoepithelial cells, and so on. And the beauty of the approach is that you can gate on these structures, such as the bronchoepithelial cells and so on, or we could use the majority cell types within those regions of interest to profile. And so we could overlay the RNA-scope data with the multiplex IF data, and then liberate the transcript data, and then interrogate the different cell types and what they look like. So this is what it looked like under the microscope. And so we could then, using CyberSort X, we could see going across here in the red is COVID, blue is normal. This is just doing a cell decon on the different cell types within the regions of interest. We can take this a step further and do an immune cell decon to look at the immune cell subtypes within each of the regions of interest within this tissue. And this is just annotating that a level deeper. And so when we look at a dimension reduction analysis, sort of a helicopter overview of the transcript data, this is at 1800 plex RNA. We can see COVID in the blue, H1N1 in the red. This is the 2009 pandemic, and yellow is the normal. And we can sort of see these two different populations. So we see this population on the right and the population on the left. At the transcript level, we can't really make out what this is. And so what we tried to now do was to add in the cell type annotation for this data set. So this is not coloring it by individual patient samples here, where an individual color represents an individual patient. Again, you can't really get much insights into this. Where it got interesting for us is when we overlaid the pathology annotation for the majority cell type within each region of interest over the tissues, over the transcript data. That's when we could see the separation of the signal. So we could see the bronchoepithelial cells were at the bottom left, the hyaline membrane was in the yellow, the type 2 pneumocytes were in the light blue, and the macrophages were in the brown. And so this is really showcasing you how you really need to integrate your pathology annotation with your spatial data to really understand what's going on. And so then we could compare the differentially expressed genes between COVID and control, COVID and H1N1. At the time, again, my background's cancer, so very limited knowledge in the infectious disease sort of knowledge base, but we had the right virologist and immunologist on the call. Kirsty Short, who's a sort of a national lead on efforts in COVID-19, was on this call, and IFI27 was one of the top differentially expressed genes between COVID and control, COVID versus H1N1. And she sort of shrieked at the call at the time on the Zoom call, and it was really interesting because it was IFI27 is known to be elevated in the blood of COVID-19 patients, but no study had tied that data to the tissue directly. And so this was one of the first studies showcasing that this type 1 interferon was highly enriched for in COVID-19 specifically. And you could see that in the H1N1 contrast as well. And so when you look at the viral load versus, this is scoring viral load by RNA scope, where you have high viral RNA versus low sort of viral RNA between the groups, you can see this type 1 interferon enrichment again. And so what we did was we took our gene signatures and we compared them to publicly sort of published studies at the time, and we could see that this IFI27 Ly6E signature was enriched for in multiple studies at the time. And so we published this work in the European Respiratory Journal and sort of piqued our interest into, we could see this signature at a multicellular readout based on spatial, and it sort of piqued our interest into whether we could apply this and understand where the signal was coming from at a single cell level. Can we identify the cell type where the cells are, where the signal is coming from, this type 1 interferon. And so we use this nanostring spatial molecular imager, which allows you to do single cell and subcellular profiling of the tissue. And we ran 1,000-plex RNA single cell readout on an independent cohort of tissues. This was collected across 40 new or 40 additional autopsy studies of lungs taken from COVID-19 patients, and we made a tissue microarray of it. And the fields of view that were used to capture these areas is shown in the yellow boxes. So we had COVID TMA-A, COVID TMA-B, and a normal control that we used to contrast this data to. And so we could then look at the same sort of approach that we used at a multicellular sort of readout. We could take the RNA scope data, overlay that with the multiplex immunofluorescent data, and then look at the transcript level readout using this approach. And so what we could see is this is now just digging down into a single core from a single patient. We've got the RNA scope in the top left, the multiplex IF image in the top right, and we can now cell type based on, I'll go into that in the next slide, where we can cell type and look at the individual cell types within this tissue. You are using 1,000 transcript sort of readout. So when we have consensus across the cell annotation algorithms, that's when we call these cell types. But what was really interesting is we can now look at individual transcripts within individual cells. And this is looking at 3 transcripts out of 1,000 within those tissues. This is taking that border and just looking at it under the microscope using the SMI technology, and we can start to map the transcripts in space. And so what you can see in yellow is CXCL10, IFI27 in red, and CD31 in the purple. And this is that border that you can see at the top left of each of these whole cores. And so we can take these and map these back onto the tissues and do some data normalization on this, and we can start to look at the individual cell types. So we can start to look at the fibroblasts and macrophages and so on when we project this onto a UMAP. And then we can start to compare between viral high and viral low, or COVID versus normal, what that signal looks like on the tissue. So this is just mapping those cell types back onto the tissue. And so when we contrasted control versus COVID, we could see that IFI27 was one of the top differentially expressed genes, specifically enriched for in the macrophages. And this was again evident to us in viral high versus viral low when we incorporated the RNA scope readout and used this as a sort of a pixel measurement across it. So we could localize that IFI27 signal using a multicellular approach, and then we could drill down into the cell type and then identify in the macrophages that where the signal was coming from. We then took IFI27 and we wanted to see what it looked like in the blood and nasal swabs of COVID-19 patients at the time of diagnosis. So this is within the first 3 days of symptom onset or presentation to clinic. And And we could see, and the study was run from Brazil, Chile, the US, Iran, Australia, Singapore, and Denmark, and in a pre-Omicron setting. And we could see that IFI-27 specifically in the blood was predictive of disease severity. And so elevated IFI-27 within the first couple of days of symptom onset was a predictor for worse outcome, ICU or ventilation status, and it outperformed other sort of clinical assessments such as lymphopenia, CRP, age, comorbidities, and symptom scores. So we published this work earlier this year, and the assay is now being developed by a molecular diagnostics company. And so it shows you how you can go from a tissue-based study into a blood-based study using those signatures. We did look at it in the nasal swabs of these patients. There was a trend for IFI-27, but it wasn't as predictive as the blood. And so we're doing a bit more of a validation there too. And so if I go into the COVID-19 cardiac study, this is looking at COVID, same approach, tissue microarray, COVID-19 tissues prepared on 1 TMA, and then we had the 2009 H1N1. We initially went into this work thinking that your inflammatory signals is going to be through the roof in the cardiac tissues. COVID and flu are likely to be the same in how they affect the heart. That was sort of a naive interpretation, but we knew that in some cases you do see really healthy people, of young age that develop cardiac complications, and so still piqued our interest as to why that was happening. And so these were tissues taken at autopsy, again severe COVID-19, and then compared to severe flu. The challenge is, how do we dial this back in a post-acute setting, and then that's some work that's ongoing. And so we use this nanostring whole transcriptome approach to understand the different structures within the tissues, and then do some differential expression analysis. And this work was published earlier last year. And so what we saw, to our surprise, was that COVID-19 and flu looked quite different. In the cardiac tissues, we couldn't identify viral presence in them by RNA-scope, but we could see transcript differences between the groups. And what we could see specifically, if I go to the VISI plots, so this is not taking your sort of Go enrichment terms and plotting it, we could see that DNA damage and cell cycle type pathways were enriched for in COVID-19. So they were upregulated in COVID-19 versus flu. Whereas when you looked at flu, the pathways that were upregulated compared to COVID-19 were your interferon and your complement pathways. So COVID-19 in severe COVID-19, when we compare it to flu, it looks quite different. It is a small study, but it showcases how these tissues are quite different in that your interferon signaling is suppressed in COVID. You've got this sort of DNA damage, and cell cycle type pathways activated in COVID-19. And so it sort of piqued our interest as to why we're seeing these markers or pathways that are typically activated in chronic disease in COVID-19 specifically. We then went on and stained the tissues using broad DNA damage markers, using gamma H2AX and a few other markers at the protein level, and we could see that staining was specific for the COVID-19 cause, and not the flu, not the 2009 flu pandemic cause. So again, a pinch of salt. This is at severe stages of disease in an unvaccinated population. And so this work was showcased around the world in a number of studies. And so I'll go on to the last study before I conclude this talk. And so we looked at age-matched placental tissues taken at between COVID-19 patients and age-matched normal placental samples. And we went in looking at the multiple layers in the tissues. We couldn't find virus by RNA-scope in these samples. Again, similar to what we saw in the cardiac tissues. We could only identify virus in the lungs. They were absent in the cardiac and placental and brain tissues. So we think that it is very much localized to the lungs, whereas you are getting the systemic sort of inflammatory response in multiple other organs. And what we saw when we compared COVID versus normal in the placenta, we could see this enrichment for preeclampsia-like signaling, specifically in the COVID-19 positive pregnancies. These were third trimester positive pregnancies. And so we're digging into this a lot deeper. A lot of pathways such as nitric oxide, and a few preeclampsia-like pathways were highly enriched for in the COVID-19 population specifically. And it goes to show you that, even in distant organs like the placenta, we are seeing differences between COVID and normal, even in the acute setting. And so with that, I'd like to thank the collaborators on this work, the patients that have given their samples and tissues for these studies, the funding agencies, and it takes a large team to put these studies together, and everyone that's contributed to these studies. And I welcome any questions. Thank you.
Thank you very much for the fascinating presentation, Dr. Kulaseha. As a reminder to our audience, if you have any questions or comments, please submit them to the Q&A portal to the right of your screen. So, Dr. Kulaseha, so in your cardiac studies, you studied patients that died from COVID. I would assume that these patients have a lot of underlying conditions such as heart diseases or diabetes. So how do you make sure, like when you see these gene expression changes resulting in DNA damage, how do you distinguish them from the underlying conditions, or is a result of the virus?
Yeah, no, it's a great question, and it's something we've been battling with for the last couple months. The challenge is when you get these tissues at rapid autopsy, there are comorbidities associated with all patients, right? Even in normal. So to be able to curate, and we try to select as best as possible. The timing of these studies were really early on in the pandemic, so unvaccinated patients. So we were dealing with what we had at the time, matched for as good as we could at the time. But what we are seeing at a broad level are trends towards differences between COVID and flu, COVID and control. And what we're trying to now do is dial these back down into the post-acute setting and seeing whether these markers are markers of long COVID and so on. So if we know that DNA damage is specifically enriched for in patients that do poorly, then we can potentially use those to then go and look at them in the post-acute setting. Again, I completely agree with you. We need to validate a lot of these findings in larger studies. It is a challenge to get autopsy tissues from these patients. So there are some complexities in it, and we try to control it as best as we can in the study design.
Thank you. And also, what's fascinating to me is that the difference in the expression of INF27 or IFI27, sorry, interferon in lung tissues, heart, and placenta. So you mentioned that there's interferon is enriched in lung tissue, but you don't see it overexpressed in the heart. I'm wondering what would be your hypothesis of the underlying mechanisms of this?
You're asking all the tough questions, No, it's a great question. Again, I think so IFI27 is a type 1 interferon. We saw that that's elevated within the first couple of days of infection. We think it's a pan-viral marker for poor disease onset. So if you have that peak really early on, we think those patients do really poorly. We know that in pneumonia, it can differentiate between viral and bacterial pneumonia. In HIV too, it appears to peak really early on. So there are associations with how IFI27 is in the cardiac study specifically. We went in thinking that your inflammation is going to be through the roof in both conditions, but it wasn't. It was it was suppressed in COVID-19, again severe, but all your sort of DNA damage type pathways were upregulated in COVID. And so it was COVID appears to affect the heart quite differently. What we're seeing is, I think what we're seeing are sort of early signs of why this is. clinically what it means, we're not sure. So we're trying to dig into that a bit more, trying to get some resolution on why these pathways are enriched for, because we know that sort of DNA damage type pathways are enriched for in cancer, diabetes, neurodegenerative diseases, and so on. So it's not clear to us at this stage why we're specifically seeing those pathways. Again, these studies need to be validated in larger cohort studies, but I think they're giving us early clues as to why COVID is potentially very different from flu.
Is IFI27 upregulated in the placenta too?
No, we didn't see it elevated in the placenta. It only seems to be elevated in the lungs of these patients, and then we can also track that in the blood. It wasn't specifically enriched for in the placenta or the cardiac tissues, no.
Okay, so I think there is a protective mechanism within the placenta that newborns are COVID negative. Do we have any clues why that would be the case?
I don't, but I'd love a placental expert to give their opinion. But again, we've used an approach and we've applied it. We're hoping the community comes in and helps us sort of decrypt a lot of this data that we're finding. The placenta is a lot more complex because you do have the multiple layers within it that, depending on where you've sampled, whether it's towards the maternal end or the fetal end, so what we want to try to do in the next couple of months is try to do a 3D reconstruction of these tissues and look at the sort of gene expression through that, so we can get an appreciation for where it is actually signaling through. But yeah, there are complex organs, and we are sort of giving snapshots here.
Thank you. You also mentioned in your talk that right now there is a direction towards spatial proteomics. So I'm wondering, have you used proteomics tools to study cancer or COVID? Have you seen any, differences between transcriptomics and proteomics, the results between the two?
Yeah. So we've used spatial proteomics in the placental study. I didn't show the data today. That was to cell type the placental samples. We do see clear differences in the composition of the tissues based on the single cell protein readout and sort of the cellular neighborhoods that are being formed within the COVID-19 placentas versus those from the normal. But the data isn't mature enough for us to sort of conclude and present that at this stage. But we are using a lot more spatial proteomics. I think it's a lot closer to pathology. It's a lot closer to the clinic. It's a lot easier to understand those data. So yeah, we certainly are using a lot more proteomic-based approaches now.
Okay, thank you. So one more question. Can you share with us what's next for your research, particularly for COVID research?
Yeah.
I know there's like long-term effects of COVID. Are you trying to identify, therapeutic targets that could potentially?
Yeah, great question. Of course, we've taken these tissue-based findings and we're trying to see, can we identify a signature that we will now track in the post-acute setting as a marker for long COVID. We are trying to incorporate studies where you have multiple different viral strains and seeing if you've got any differences in the transcriptional profiles or the proteomic profiles of those different variants. We're doing a bit of work there. The infectious disease communities reached out to us with 100-year-old Spanish flu samples. We're about to embark on that study in about 3 weeks. These are Spanish flu samples that have been collected from people that died in the sort of 1920s in the Arctic, total bodies preserved in museums. And so we've been able to access some of those incredibly, well annotated samples as well. And so we know that, the virus is present and we're trying to do some transcriptional and protein profiling of those tissues, which I think it's opened up a really exciting sort of field of research in the infectious disease community, because typically they didn't really profile tissues, and now the technologies have caught up, so you can. So I think there's a lot of activity for us in the COVID and infectious disease work going forward.
Fascinating, thank you. Can you also share us with little bit about your cancer research. What's next for your cancer research using spatial biology?
Yep. I think some of the best data we've seen to date is the spatial metabolic data. It's really powerful in that we can see there are signatures that are predictive of disease, of sort of poorer outcome based on metabolic profile within the tumor compartment specifically. What we're trying to now do is develop panels of markers that we can screen tissues en masse at the time of diagnosis, and potentially prospective studies that we can predict, which patients will do well, which patients will do poorly to immunotherapies. And we're doubling down on a lot of the high-plex protein profiling approaches, and using that data to start to impute into H&E imaging what that looks like at a feature level. And I think that sort of democratizes a lot of spatial into the AI space and digital pathology space. I think a lot of the work we do in cancer lends itself into the immunology infectious disease spaces. But yeah, the metabolic and epigenomic data, spatial data is really, really powerful at the moment.
Fascinating. Thank you. Okay, that's all the time we have for today. If you have any further questions, please consider reaching out to the speaker directly. His email is shown on the screen. As a reminder, the webinar will be archived on the DDN website, and you will receive an email notifying you that when the webinar is available on demand. On behalf of DDN, I would like to thank our speaker, Arutha Kulasinghe, as well as sponsors, Sino-Biological, Canopy Biosciences, 10x Genomics, and Fortis Life Sciences. And of course, our thanks to you for listening. Goodbye and have a good rest of the day. Thank you.